Hello everyone, thank you for your time for the lightning talk. This is Chao Zhang from Dr. Li Boshan and in his lab in Department of Plant Pathology and Microbiology, Texas A&M University. Today I'm going to talk about MPK3 ubiquitination dynamics in plant immunity. Plants protect themselves from pathogen infection through multi-layer defense strategies, including preformed defenses, induced defenses, which is the focus of our study, and systemic defenses. During pattern-triggered immunity, conserved pathogen-derived molecules as elicitors, called PAMs or MAMs, can be perceived by cell surface receptors. For instance, here, the receptor FLS2 can recognize bacteria flagellin and recruit co-receptor and activate downstream immune responses, including MAP kinase cascade, ROS production, and so on. Also, the immune responses need to be dampened off to avoid over-defense. So how is plant immunity regulated? Studies have shown that post-translational modification, including phosphorylation and ubiquitination, plays an important role in this process. Ubiquitination, the study of which is still at its infant stage, attracts our attention. Ubiquitination is the process of addition of ubiquitin, a 76 amino acid protein, to the substrates through a three-step reaction catalyzed by three enzymes. It can be classified into monoubiquitination, multimono, or polyubiquitination, and also different linkage types of polyubiquitination based on which lysine residue is used. Then, how does ubiquitination function in plant immunity? To answer this question, first we would like to isolate ubiquitinated protein during plant immunity. So we use this two-step affinity method to purify ubiquitinated protein. For ubiquitin-associated domains, which can bind ubiquitin connected head to tail, termed tubes, was used to isolate ubiquitinated proteins. Abstract of wild-type or histidine transgenic Arabidopsis seedlings were applied to tubes affinity column and washed and released, and then subtracted to a second step Nico affinity purification. From this anti-ubiquitin Western blot, we can see that this method can effectively enrich ubiquitinated protein. And the conjugates was then analyzed by mass spectrometry, and hundreds of ubiquitinated proteins were identified and shown here as this interaction network, including the components in ubiquitination and defense-related components. To further confirm the identified proteins are indeed ubiquitinated, we detect the ubiquitination of the candidates by in vivo ubiquitination assay in Arabidopsis protoplasm. From all the candidates we tested, we can see the ubiquitination of MPK3, a single kinase in MAP kinase cascade, was clearly enhanced in 10 minutes, shown here as a single band, and well decreased in 30 minutes. We did not see clear changes of other candidates. Since MPK3 and MAP kinase are important components in plant immunity, next step we would ask what's the role and mechanism of MPK3 ubiquitination in plant immunity. We would like to test if MPK3 ubiquitination involves in plant immunity, and to identify the E3 ligase and the ubiquitinase for MPK3, and to test the other possible outcomes of MPK3 ubiquitination. Ubiquitination and deubiquitination has been proved to be important roles in regulating human immunity through a variety of E3 ligase and deubiquitinase, which show a high similarity in plant immune pathways. Study of how plant immunity is fine regulated would be helpful for crop improvement and disease control in other species. That's all for the talk. Thank you.